This week we're going to be examining the topic of biotechnology, and biotechnology has come to mean a lot of different things in the world of science, both from the standpoint of allowing us to better understand the world around us and human physiology, but also in terms of allowing us to manufacture crops, for example, that are resistant to different viruses or to insects, to manufacture animals that will make things for us like insulin. And even doing goofy things like you see in this picture here, where just to see if we could do it, we manufactured a mouse that glows in the dark. And yes, it does. Just it has the same protein in it that fireflies do that allow them to glow. So what is biotechnology in the first place? Well, basically biotechnology is applying the knowledge that we have of biology and the techniques that we've developed to study biology to enhance human life, human medical practices, forensic studies. Forensics means or refers to the use of science in the courtroom, and to also enhance our industries and agricultural yields, etc., all using what we understand about biology and the techniques that we've developed through the study of biology. Now, there's another related term called genetic engineering. Genetic engineering is any time you manipulate a DNA sequences for your purposes, such as, for example, manipulating a genetic sequence in order to make a plant resistant to a particular virus. Frequently, genetic engineering will also lead to the production of what's called recombinant DNA. Recombinant DNA is any time I take DNA from two different organisms and combine them together. So if we go back to the example of our glow-in-the-dark mouse here, that mouse represents an organism with recombinant DNA, because along with all of the normal mouse DNA you expect to see inside this organism, we also now have inserted into the mouse chromosomes the gene for the manufacture of a protein that allows it to glow in the dark, and that gene came from the firefly. I'll give you another example of recombinant DNA, and this is one that is very common today in industry because it's of primary importance to diabetics, and that is we have been able to manipulate bacteria so that they will manufacture human insulin for us, and then we can take that insulin, purify it, and make it available to diabetics that cannot produce their own insulin. The way that we do that, this picture shows in pretty good detail, but I'll just tell it to you very quickly, is we insert into the bacterial chromosome a region of DNA that codes for the human insulin gene. And once that has been successfully incorporated into the bacterial DNA, that bacterium will be fooled into making insulin, thinking it's just one of the many different kinds of proteins it's supposed to make during its normal bacterial life. We can then take that insulin and, again, as I said, purify it and make it available to diabetics. We use the same procedure to also make human growth hormone, or HGH. These are just two examples of recombinant DNA. Now, another example of another technique used in biotechnology is something called the DNA fingerprint. You're probably most familiar with this from watching episodes of CSI or from listening to courtroom testimony that discusses whether a certain suspect's DNA fingerprint matched the DNA that was left at the scene of the crime. So DNA fingerprints are frequently used in forensics. Again, forensics is the application of science in the courtroom. And a typical DNA fingerprint would look something like this. This is a case where they used DNA fingerprinting to establish the paternity of children, in other words, who was the father. And you can see that in this region right here, the mother, this lane represents the mother's DNA. Here's each offspring's DNA. Here's the father's DNA. You can see the mother does not contain a certain section of DNA, which is found in both of her children and also found in the putative father. And so as a result, we would say that the only way that these two kids, these offspring, would have this particular band of DNA present in their DNA fingerprint is if it was donated to them by their father, since their mother does not possess it. So we can use DNA fingerprinting again in the courtroom. We can use it for paternity determinations, and evolutionary biologists like me can use it to establish relationships between organisms. Let's say, for example, we want to figure out how closely related dogs are to wolves, or dogs are to coyotes, or coyotes are to wolves. We can use the same kind of DNA fingerprinting technology to determine the closeness of relationships between organisms. So how do you go about creating a DNA fingerprint? Well, first of all, you need to have some DNA. Now, that doesn't sound like so tough, but imagine for a minute that let's pretend that you're a detective and you are investigating a crime scene, and a single drop of saliva was left at the crime scene. That single drop of saliva will not contain enough DNA in it for you to perform the kind of tests you need to determine who donated that DNA, who left it at the scene of the crime. So the first order of business once you have this DNA is to make sure you have enough copies of the DNA in order to perform the tests you want to perform. And the only way that you're going to be able to take that small amount of DNA and make lots of copies of it, in other words, amplify it into a much larger amount of copies, is through a process called PCR, which stands for polymerase, as in DNA polymerase, chain reaction. So let's take a look at PCR and find out exactly how we do this. Now, the cool thing about PCR is this is a technique that was developed in the 1980s, and today we can perform this entire process of copying DNA much like you copy pages out of a book using a copy machine. We can perform this entire thing in a tiny little machine like you see here that can sit on the top of a lab bench. In fact, we will show you a PCR machine in class, and you'll get to actually use a PCR machine in a week or so. So the first thing you're going to do is you have your DNA that you've collected, and you are going to add into this PCR machine not only the DNA that you have collected, but into the same little bin where that DNA is, you're going to add some primers that are going to adhere to the regions of DNA you want to copy. Remember that DNA polymerase cannot copy DNA unless there is an RNA primer first attached to the DNA, so we're going to need those primers to initiate replication. We're going to add in nucleotides, that's A's, C's, T's, and G's, so that they can be used to make a copy of the DNA. And then we're going to use a special type of DNA polymerase called Taq polymerase. And Taq stands for Thermus aquaticus. Thermus aquaticus is a particular bacterial species that is actually an archaea, which is related to bacteria, but it's one of the prokaryotes. It lives in an extremely hot environment in the thermal pools found in Yellowstone National Park. So this bacterium tolerates exceptionally high temperatures of on the order of 70 to 90 degrees Celsius, near boiling. Why do we use Taq polymerase and not just regular old DNA polymerase? Well, you will see here in a second when I show you the process of PCR that one of the things we have to do is heat the DNA up pretty high to get it to denature and split apart. And when we do that, if we use regular old DNA polymerase, for example, our own DNA polymerase, the DNA polymerase would denature at the same time the DNA does. However, Taq polymerase comes from an organism that's used to existing in a very hot environment, and thus it has developed a type of polymerase that does not denature at high temperatures. So it still remains functional when we heat up the DNA to denature it. So we've got all this stuff added into the chamber, and we turn on the switch, and the copying procedure begins. And here's pretty much what it looks like. So up here in step one, we've got our DNA that we've collected, and we've added not only our DNA into the chamber, but remember again our individual nucleotides, the adenine, guanine, cytosine, and thymine. We've added in some RNA primers to start the process of copying, and we've put in that Taq polymerase. The first thing that we're going to do is heat up that DNA and all of the reagents in there to the point that the DNA denatures. That causes the hydrogen bondings between the nitrogenous bases down the middle of the DNA to split apart so that we get two separate strands, as you can see here. Then we cool it just slightly, just enough cooling, so that, as you can see here, our RNA primers will adhere or stick to the complementary regions of DNA. And once that happens, now Taq polymerase is going to come in and start actively synthesizing new strands of DNA to make a full double-stranded DNA again. So that at the end of one round of the PCR reaction, we will have double the number of DNA that we had before. If we repeat the round again, we'll have double again, or 8 times as much. So the more times that we repeat this cycle, going from 6 back to 1 and repeating it again, denature, cool it slightly, allow the Taq polymerase to make the copies, and then heat it again to denature the new copies, cool it slightly for the primers to anneal, allow the copying to go again. The more times we keep repeating this, the more copies of DNA that we get, such that by the time we get done with about 30 cycles, we should have roughly 1 million copies of the DNA that we're after. And once we have 1 million or so copies, now we can finally start using that DNA to do other things, such as determine if the suspect's DNA matches the DNA found at the scene of the crime. You can also use forensics to identify infectious agents. Let's say there's a new strain of flu that's appearing. We want to know whether or not individuals in a region of the country have contracted that particular strain of the flu virus. Again, we're not going to have very much of the flu virus that we get from a patient. We're going to need to first amplify the DNA using PCR before we can run tests to match it with known strains of flu. We use this same process in the Human Genome Project. That was a project by which we sequenced or figured out the orders of A's, C's, T's, and G's on all of the chromosomes found inside of a human cell. We can use it in taxonomic and systematic studies, in other words, studies that are establishing the relationships between organisms. And medical researchers sometimes use PCR as a starting point for their studies of cancer and other types of human diseases. And that's the end of part 1 of the biotechnology